Now we're going to move on to the digestive system. And if you look at the whiteboard over here to the left edge of the left bank, you see something that looks very familiar. This was also on the whiteboard for lecture exam 1, and it's the 4 biologically important macromolecules. Basically, we're going to talk about several terms that lay people, patients, just take for granted. They are specific terms that have specific meanings, and as a healthcare provider, you need to know what these specific terms actually mean. For example, digestion and absorption are 2 different things, 2 different processes. They are not the same thing. And when we talk about digestive basics, so carbohydrates, which is the primary energy source for cells. We have these huge polysaccharides that some enzymes are going to break down into 2 links, disaccharides, and then you'll have other enzymes breaking it into single link monosaccharides. And the monosaccharides are what is absorbed. For proteins, we have proteins being broken down by some enzymes into shorter chains, and then those shorter chains being broken down into individual links, which are amino acids. Now, out of all our proteins, we only use 20 amino acids. Our body has the enzymes to make 12 of those 20. 8 we cannot make. We must eat those amino acids. We call those 8 amino acids essential amino acids. And then lipids, yeah, it doesn't work quite as well, but lipase will break down triglycerides into 3 fatty acids. Actually, we're going to see that that's actually not true. It's 2 fatty acids. And glycerol, and actually it's a monoglyceride. Again, a lot of things that I cover in this class has been oversimplified, and I'm going to add in a few of the real complications in this lecture and next lecture. All right, well, the top 3 are, 1, 2, and 3 are the nutrients. We need to eat a lot of them because there's a lot of those macromolecules in our cells. But our digestive system will break down nucleic acids, and we will actually absorb nucleotides. It's not essential, and you don't get very many, but our body will actually make enzymes to break those down as well. Now, if you look at on slide 2, the top 3 diamonds and the boxes, and the green named enzymes on the left of those 3 boxes, those were the 3 enzymes of lab 3, the group lab report lab. Front table had amylase, middle table had pepsin, back table had lipase. Okay, now, what I want you to get out of this is that all these catabolic reactions on this slide, that is digestion. Write this down next to slide number 2. Chemical digestion includes all the catabolic chemical reactions that are breaking the 4 biologically important macromolecules into individual monomers, and those individual links is what we absorb. But getting to the links, that is digestion. And why do we have to break them down to single links? And it's because that size matters. Okay, only the single links are small enough to be absorbed. Now, when we talk about that, I have mentioned this before, but this is really important, especially when I get talking about the pancreas, that you understand this concept of the digestive tract, like our bodies being glorified donuts. And if you have a donut on the table in front of you, the donut hole facing the ceiling is your mouth. The donut hole facing the table is your anus. And the whole donut hole is your digestive tract. And when we cover the digestive system at these levels of classes, we tend to split it up into two main sections: the digestive tract, alimentary canal, and then the accessory organs. I'm going to kind of cover them both at the same time, but that's how we tend to divide things up for introductory students to help understand the digestive system. But what this slide 3 is talking about is we got to get these huge chains, which, if they stay huge, are going to stay in the donut hole. And we got to break them down to individual links, because those are small enough to bring into the dough. If it is in the donut hole, if it is in the lumen of your digestive tract, it is outside your body. If you want to be in the donut, you got to be in the dough. Okay? All right. Slide 4. This is just from your textbook showing you how these enzymes break these large chains into links, and it's not done all at once. Again, it's the first enzymes will break them into smaller chains, and then those smaller chains are broken up into individual links. All right. Now, the digestive tract lining, what's lining the donut hole, is by far and away simple columnar epithelium and loose connective tissue we call lamina propria. Add those together, we call that tunica mucosa. Okay? Simple columnar epithelium, lamina propria equals tunica mucosa. For the most part, an average digestive tract, average size adult is about 30 feet. Okay, that's a long donut hole. And of that donut hole, probably 26 to 27 feet is this simple columnar. There's a little bit at the beginning and just a little bit at the end that it stratified squamous epithelium, but the vast majority of it is this that's labeled here on slide 5. Okay. Now, if these enzymes are designed to break down the 4 biologically important macromolecules, and your cells are made up of the 4 biologically important macromolecules, your body has to have protective mechanisms to keep your body from digesting its own donut hole. Otherwise, you're going to digest the digestive tract. And so to protect the tunica mucosa, there are various mechanisms that your body has. One is protective mucus. Protective mucus can come from either goblet cells, and that's what the G's are on this picture, or mucus neck cells, which are specialized. They're not goblet cells, but they are found in the stomach, because the stomach has really acidic acid, and you need to be able to protect the stomach much more so than even other areas. And then, again, the cells that are making the digestive enzymes do not make digestive enzymes. They make the inactive precursors to the digestive enzymes. And that way it doesn't digest the cell that's making it. We talked about this a little bit in Lab 3, about how in the stomach, it's not pepsin that is made, it's pepsinogen, an inactive precursor. And then once it's secreted into the donut hole, into the lumen, wherever that is, stomach or small intestine, there's a combination of ingredients in the lumen that activates those inactive precursors to things that will actually break down chains in the links. And number 3, these cells, these simple columnar epithelial cells, have a pretty high rate of mitosis, of cell cycles. So there's a high turnover rate. For example, on the small intestine, you guys remember intestinal villi. The stem cells are down there in the pits between the villi, and they're constantly dividing, and all those simple columnar epithelial cells are migrating up to the top of the villus, and they're shed at the top of the villus. And the rate of that cell division and migration is almost perfectly timed. By the time that simple columnar epithelial cell sloughing off the top of the villus, it is about ready to become digested. Okay? And this is also a thing, we don't see it too much anymore, but back in the old days of chemotherapy for cancer, therapeutic agents, most of them are targeting cells that divide quickly. And so that's why people would lose their hair, because the cells in the hair bulbs are dividing quickly and people lose their hair. Well, cells of the digestive tract are dividing quickly too. And in the old days, we used to see a lot more of this. We still see it, but not nearly like we used to. But we would see peptic ulcers from people who were in chemotherapy because these cells were getting targeted. Okay. Slide 6 is a picture of the donut hole and the various linings. Remember, it's a 4 layered digestive wall. That's from anatomy. All right, so let me do the bottom line, the big picture. The whole issue about digestive physiology is about break down as many chains and the links as possible, and then absorb the links into the dough as much as possible. Anything that stays in the digestive tract lumen, it remains outside your body. Okay? That's the top slide 7. All right, let me move over here. Okay. Now we're going to basically do a little trip through the digestive tract, starting at the donut hole facing the ceiling, and we're going to go all the way down to the donut hole facing the desk. I'm going to be talking about accessory glands, some of the accessory glands along the way, but there are various processes that are going on, and I have 3 of them that are relevant for today's lecture, and then we'll have more later. But in purple are the words that apply to processes of mechanical processing. And then if I underline it in blue or star it in blue, that has to do with secretion. So your body is actually putting a lot into the donut hole, and then it's going to take out a lot of the donut hole into the dough. But they're 2 different processes. And if it's blue, that means we're putting it into the donut hole, into the lumen. And then when we get to absorption, that's where we're going to take it out of the lumen and into the dough. Okay? All right, so the process of ingestion is basically putting things in your mouth, and it is guided by the hypothalamus. The hypothalamus is a crucial area of the brain. There are certain areas that are what we call feeding centers, and it's thought to be in these areas of the hypothalamus, lateral and ventral medial. But the hunger center is going to give you the feeling like I want to eat. And there are various things that trigger hunger centers. And there's also satiation centers that kind of give you like, I've eaten enough, I need to stop. A lot of actual eating disorders have to do with issues with these centers. Now, not just organic, there's a problem in the hypothalamus, but there is a lot of behavioral things that influence these hypothalamic centers. But when people have eating disorders, whether that is chronically overeating or if it's like anorexia, it's things that are affecting the way these centers are working. Okay? And all these things are working in balance to give you a healthy appetite. Healthy doesn't necessarily mean you're always hungry. Healthy means it's balanced so that you can eat what you need and not go beyond that. Okay? All right. You don't need to know the name of the actual hypothalamic nuclei. Just know that there are hunger centers and satiation centers. So there's a couple slides here that talk about some things that can influence those centers. One that influence the satiation center are stretch receptors in the stomach. And if they're stretched, they'll send more signals to the satiation center, and that gives you a feeling like you're full, I can stop eating. Interestingly, these stretch receptors are sophisticated enough to tell the difference between whether you're stretched by more solid food versus just liquid. I'll talk about that later when I get to water and electrolyte balance. Blood glucose levels will also influence appetite. If you have lower blood glucose levels, it'll signal or stimulate your hunger center. If you have higher blood glucose levels, it'll stimulate your satiation center. Obviously, from Lab 8, if we have poorly controlled diabetes mellitus, that gets all messed up. Okay. One of the things, and you may have noticed, number 3, if you get cold, you tend to get hungry. And one of the reasons why, I wonder if anybody here can give a guess why it is that if you get cold, you'll tend to get hungry. And the hint is shivering. Sliding filaments is what gives us our core body temperature. If you are cold, that's why you will have an involuntary reaction of your voluntary muscle to shiver. You get more sliding filaments, and that increases your body temperature. While not all sliding filaments are in skeletal muscle, you also have sliding filaments. They're definitely more diffuse, and that's why you don't see striations in smooth muscle. But smooth muscle, when it's contracting, will also generate heat. And so if you get cold, you will get stimulated hunger centers to eat. You'll do some peristalsis, some stomach churning, some segmentation, and that'll help heat your body up. Number 4, this is slide 9. You can have specific cravings. And some of them are also, you may not be craving it, but you better learn about this because you can use this to your own advantage when you are in your programs. For example, on point number 4, if you know that if you eat simple sugars or carbohydrates that are easily broken down into simple sugars, you will get a spike of blood glucose, and it'll come right back down. This is Lab 11. I gave them all the volunteers, Lab 8 that we didn't do. But when the volunteers are given glucose solution, those are just given single-link glucose. Blood glucose goes like this, and boom, within a couple hours, it's coming way back down. And there is such a thing called reflex hypoglycemia. If you have too high a sugar, and that happens when you ingest sucrose or glucose, it comes right back down, and it's even lower than what you started. But if you remember, like on what it's saying on point 4, that if you eat protein, which will get broken down into amino acid, that amino acid is going to go through gluconeogenesis, and that's a lot slower. And you'll tend to get less of a peak of blood glucose, but more of a slow, sustained rise of blood glucose as gluconeogenesis gets rolling. What does that mean for students who want to go into programs? You got an important class, or you got on Wednesday lecture exam 3. Don't eat a bunch of Lucky Charms or Cocoa Krispies before lecture exam 3, because before it's over, you're already crashing. Instead that day, have some eggs, have something with protein, and you'll get a more sustained increase of blood glucose. Your brain will have adequate glucose because it's so sensitive to blood glucose during the whole exam. When you get into taking longer exams, this is going to be even crucial. The DAT for me was 8 hours. Part 1 of the National Board exam, 8 hours. Part 2 National Board exam, 2 8-hour days. And if I didn't understand this and actually apply it to my own life, I wouldn't have scored as well. We are learning about the human body. Use it to your advantage. Okay? All right, and then general nutrition state, again, and this is, your body can actually give you clues about what you need. You'll actually start to crave things that you are deficient in. Generally, by the time you're at this stage, you should be able to understand how to control your diet and have balanced nutrition. But what point number 5 is saying is if you have toddlers, pick your battles. And this ain't one of them. Okay, they're not eating the vegetables. Well, every now and then you got to kind of encourage them and eventually they're going to eat something that they're missing because their hunger centers are going to get triggered because they're not eating enough of this. So pick your battles. And then 6 has to do with other behavioral or psychological things that influence the hypothalamus. Some people eat out of boredom, out of habit. A lot of it is tied to the limbic system. When people get heartbroken, they get a box of chocolates, that kind of thing. And then at the very bottom right of the slide is my comfort food, and that's Mint Milanos. And what's so great about Mint Milanos is they come in that single serving bag, because I'll finish that whole bag in one sitting. All right, slide 10. Let's start going down the pathway. So you ingest it, you put it in your mouth. Now it's in your oral cavity. In the oral cavity, this food is just called food, but your salivary glands are adding saliva to it. While it's doing that, you're undergoing the process of mastication. Boy, I hope that came clearly through the camera microphone. Mastication is chewing. These animations that are on slide 10 actually came from a dental school website, and when I saw it, I brought back all these memories. I'll just give you one tidbit from these animations on slide 10. The slopes of your mandibular fossa on your temporal bone are actually paralleling the slopes of the cusps of your teeth. Like bicuspids have 2 cusps, all right? So when you have everything anatomically correct as you are chewing, the mandibular condyle of your TMJ is sliding in the same angles that are on your teeth. When they are not in the same angles, that's where parts of the capsule that encloses the TMJ and part of the dense regular connective tissue that holds the meniscus between the mandibular condyle and the mandibular fossa, it starts getting stretched out. And this meniscus kind of flips back and forth instead of staying put in between the ball and socket. It's not a ball and socket, but you know what I mean. And if it starts doing, that's where jaws start to pop and click. Now that came back to me as I saw that. So the muscles of mastication are muscles that move the mandible, and that's just the masseters temporalis, medial, and lateral pterygoids. There are only 4 pairs of muscles that actually you use for chewing. All right. Then, so I mentioned saliva, and that's our first secretion. That's why it's underlined in blue. There are things that can trigger your salivary glands to go. One is if you're hungry and you smell something yummy, your salivary glands are going to go. Also, if you just put anything in your mouth and roll it around, just the tactile receptors in your mouth and the tactile receptors in the periodontal ligament holding the teeth to the bone, you stimulate that and you're going to stimulate salivary secretion. Remember for lab 3 how I was the bull mastiff dog that supplied this saliva. I have this desktop basketball game where you flip this small little plastic basketball into this hoop. Well, I wash it every time, but that's what I do. I put it in my mouth. This piece of plastic rolls around, and I'm making a lot of saliva. Okay. Again, use this stuff to help you get the job done. By far and away, saliva is water. It's mostly water. But there are some things that are in it, and we've actually learned a couple things now. One from Lab 3 is we have salivary amylase, also called alpha amylase or talin. From the immunology lectures, we also have IgA in saliva. So there are things in saliva that's not just the water. But this salivary amylase breaks down starch into disaccharides. Again, it's got to do this in steps, not in one step. Its optimal pH is 6.8. It says here between 6 and 7. That's the top of the bell-shaped curve. One thing, though, is that chloride ion must be present in the mouth for salivary amylase to work, and it usually is. It's part of table salt. But remember, and this bottom diamond is telling you what one of your front table conclusions were for Lab 3A. Salivary amylase is relatively irrelevant. You would not miss it if you didn't have it, and that bottom diamond is showing you that. And that's because it's only present in starch for probably less than a minute from the time you put it in your mouth to the time it hits the stomach. Now this is showing you the major salivary glands. What textbooks at this level don't talk about is the entire mouth is covered with microscopic salivary glands. I think they're referred to as buccal glands, but you have a bunch on the inside of your lower lip. If you ever see this, looks like there's a bubble in the lower lip, like a blister. If you see it in your kids, calm down. It's not, oral carcinoma that you're going to have to cut off half your face. It is called a mucocele, and what it is one of those tiny little microscopic glands. The duck got pinched. It's still making saliva. You've got a little spit blister. And all you got to do is put warm compresses on it or swish with warm water, and eventually it opens up and all the saliva will go out. You don't want to have it cut out, because if you go in there and try to cut it out, you are actually pinching other little ducks, and you are at risk for making more. So just keep working it. It'll loosen up and eventually drain out. All right. Let's see. So these are mechanical processes. You got to put the food in your mouth, then you got to chew it, and then you got to swallow it. And that word for swallowing is deglutition. Here in the pharynx, starting in the pharynx and throughout the esophagus, this chewed food plus saliva is now called a bolus. So in the mouth, it's called food. As soon as you put it in the oral pharynx, it's called a bolus. Okay? And when you want to swallow a bolus, you have to lift up your tongue. And when you lift up your tongue, you push the bolus against the soft palate. The soft palate raises up, and with the uvulus seals off the nasopharynx. And then the rotation of lifting up the base of your tongue to do this rotates the epiglottis down over the opening to the larynx. This forces the food down the posterior fork, which it would be the beginning of the esophagus. So this is all happening between the oral pharynx and laryngopharynx, and starting right at the end of the laryngopharynx, at the very beginning of the esophagus, peristalsis starts. So peristalsis starts at the very beginning of the esophagus. Then these are wave-like contractions that you do not need any nerve impulses to generate. It's the stretch of the tube itself that causes it to work. And the peristaltic waves will deliver the bolus past the esophageal cardiac junction, and it plops into the chyme of the stomach, okay? Well, this is a two-way street, so while we're here talking about swallowing, we got to talk about vomiting. That's just in the opposite direction. Slide 14 is showing you a figure from your textbook showing you the sequence that I just described. All right. This is usually a defensive response to toxins. Remember, everything is toxic. So it means you have gotten enough of something to the point where it's going to be trouble if it stays in your digestive tube. So it wants to get rid of it. And the sequence is you got to take a deep breath, because for a while, you better not be breathing. By the way, when rock stars die of overdoses, a very common way that they die, you think, well, they died of a heart attack or this or that. The most common way people die of drug overdoses is they vomit, but they don't close off the larynx, and they aspirate pH2 chyme. The delicate tissues of the alveoli just instantly go poof and pH2 chyme. And that's basically how a lot of drug overdoses die, is aspirating vomit. Well, if you've been out partying, especially there's probably going to be a bunch of parties once shelter in place is lifted. If you drink too much, and this is going to happen, just make sure all your air passages are closed. All three layers of muscles of the stomach is going to contract violently. The lower esophageal cardiac sphincter is the only thing that's going to relax. And also the esophagus is going to relax. Peristalsis shuts off. When all three muscle layers of the stomach contract violently, it is a violent ejection of pH 1 to 2 chyme. The diaphragm and abdominal muscles are also contracting. It is a full body exercise. Okay? This is triggered by the brainstem. When we talked about the vital reflex centers in the medulla oblongata, I only mentioned three: cardiac control center, respiratory rhythmicity center, vasomotor center, but those are the three relevant for this class. There are many other vital reflex centers. Here's another one listed on the bottom of this slide 15. You guys like all my pumpkin slides? You can kind of tell by the time I got to this point of PowerPoint making, I was getting kind of punchy. All right, so slide 16, we're getting into the stomach. Here we have gastric glands secreting a lot of gastric juice. Your stomach has a lot of liquid in there all the time. Even it will have liquid in there when you're not eating. And this is why when the bolus goes past the esophageal cardiac junction, it plops into liquid, and that's the gastric juice. So again, that's secretion. That's what your body is putting into the donut hole. Here is where we call it chyme, okay? Now the gastric juice comes from the gastric glands, and there are various different cells in the gastric glands. The ones up there really close to the opening at the neck of the gastric glands are the neck cells, mucus cells. Again, these are not goblet cells. There are no goblet cells in the stomach. Parietal cells secrete intrinsic factor. This is important for absorbing vitamin B12 in the small intestines. If you don't have adequate intrinsic factor, you're not going to have adequate vitamin B12 absorption, and this is where you're at risk for pernicious anemia. But the parietal cells are also responsible for the ingredients of the stomach's acidity. It is secreting out hydrogen ion and chloride ion separately into the lumen of the stomach. In the stomach, that's hydrochloric acid. But the parietal cells cannot make hydrochloric acid and secrete it out to the lumen. It has to do it separately, otherwise they would dissolve from making that acid. And then the chief cells are making that pepsinogen that we learned about in lab 3. Now, what I want you to get to, that should have been covered in anatomy, most of these digestive organs, starting with the stomach, are also endocrine organs. The reason why we don't study it with the endocrine system is because these are more short loop hormones. Hormones is where one endocrine gland, in this case these stomach gastric glands, are secreting a chemical that goes into the bloodstream and causes another cell to be set in motion, hormone to set in motion. And the ones that we study in the endocrine system are going full body, head to toe. And these aren't. They're short loop hormones, but they are technically still hormones because they're going into the bloodstream and setting other cells in motion. In this case, the gastric, and the best way to think about these hormones is this whole sequence that I'm doing here on the whiteboard. Once the stomach has food and it's doing its thing and it's about ready to release chyme past the pyloric sphincter, those next organs better be ready, and that's the short loop. These hormones from the enteroendocrine cells, here are serotonin, gastrin, and somatostatin, are going to the pancreas, gallbladder, and duodenum, and it's getting that next region ready to go. And then slide 17 is showing you that picture of those. So gastric pits are the holes that lead to the gastric glands. And you can see that the neck cells are all right there, right at the very beginning of the gastric glands. That's just before it opens up. Okay, and here on slide 18, again, is what I wrote here. You got the bolus plus the gastric juice, we call that chyme. In the stomach, there is very little digestion. There's very little digestion over here at all. Let's see, what color should I use? Maybe I should use red for digestion. Use this for absorption. So we get a little bit of carbohydrate digestion from saliva. Let me see, what is that number again? Here we have very little protein digestion from the pepsin. Remember, it can only initiate it. And there's very little absorption as well. Mostly just water and salt, a little sugar, and a little alcohol. And that's why you've got to be careful if you're drinking on an empty stomach, because it goes in right from the stomach. It's not going to wait till it gets to the small intestine. And here's where you got to think about dosage and percentages. When you have an empty stomach, yeah, there's a little bit of gastric juice in there, but there's very little. When you drink, that alcohol becomes a much higher percentage of what's in your stomach. If your stomach is full with other things, you have the same amount of drink, it is a smaller percentage of what's in your stomach, and it's all kind of going into the stomach at that time, and you can spread it out if you dilute the alcohol with other things in your stomach. If it's on an empty stomach, it's going to go flying in. And that's why I have this picture on the bottom right. This guy made it to the toilet, but he forgot to take his pants off. Or this guy made it to the toilet, but he broke it. All right. Again, this is expanding on the stomach's protective mechanisms, because especially with that pH 1 to 2, you need quite a bit of protection there. Okay. So again, the chief cells are not making pepsin, they're making pepsinogen. Parietal cells are not making concentrated hydrochloric acid. It is making them separately. But when all those things go into the lumen of the stomach together, that acidity, so the picture on the bottom left is showing you how the parietal cell is secreting hydrogen ion and chloride ion separately. And then on the bottom right, you can see how pepsinogen is a larger protein that does nothing. And as soon as it gets to pH 5 or below, it loses the first 44 amino acids, where methionine is gone. It's no longer the first protein of pepsin. And that shape is perfect for binding 2 amino acids. And when 2 amino acids in a chain bind to that active site, the whole boulder changes shape and it splits the peptide bond. Okay? So this is showing you how in your stomach you're going to have much more mucus. The mucus cells secrete a lot more mucus than goblet cells. And all these ingredients are inactive until they are mixed together in the lumen of the stomach. All right, slide 20, we start into the small intestines. I list the pancreas there as well. This is where in the small intestine, this is where all that is completed. See, for absorption, this is mostly just and for this is most absorption. Not all. We have a little bit in the stomach. We went more absorption here in the intestine. After the intestine, there are more diet in the small intestine. The pancreas, this is the second accessory gland we've mentioned. Salivary glands are first. This is second. This is the primary source of digestive enzymes. Okay. Now again, it's getting hormonal clues. It's getting autonomic parasympathetic stimulation. So when it's talking about the digestive system, when it says autonomic nerve impulses, this is parasympathetic. Remember, parasympathetic is ease and feast. So the autonomic division that gets the digestive system going is parasympathetic. All right. The gastric phase of stomach secretion, again, is also parasympathetic. But when it's getting that it is also getting the pancreas going. And then once acidic chyme is released from the pyloric sphincter, it causes the small intestine to release secretin as well. Again, go back to the gastric glands. This is one of the things that the gastric glands is secreting, the endocrine cells that secrete secretin, and as well as gastrin and somatostatin. All right. Again, because especially when you're looking at the small intestine, you're looking at 20 feet. And so the initial part, the duodenum is going to be releasing this, the pancreas is releasing this, and it's going to start to go downstream down the 20 feet, getting the jejunum ready and getting the ileum ready. This is slide 21 is a reminder of the anatomy because it is crucial. Now, if you look at this figure on slide 21 and you see that reddish duodenum, at the very top of that tube is about the pyloric sphincter. That pyloric sphincter is the beginning of the duodenum and the end of the stomach, and you can see how close the pyloric sphincter is to the hepatopancreatic ampulla, labeled here as the ampulla of Vater, which is the old name for it. I remember in lab 3 when we're talking about the conclusion for the back table for lipase. You can see that in the common bile duct is where you're getting bile salts. In the pancreatic duct is where you're getting lipase. And I said our bodies are designed to maximize lipid digestion by delivering both bile salts and lipase at the very beginning of 20 feet of small intestine. Look at this picture on slide 21. That very top of the duodenum is the beginning of 20 feet, and right there is where it's being delivered. At the bottom of that red tube, add 20 more feet to have all this digestion and most of it being absorbed. Okay. Let's just move this over. Slide 22, let's talk about the pancreas because this is a crucial organ to your ability to digest. Again, the secretin is going to stimulate the pancreas to secrete the watery fluid with the bicarbonate ion. This is the chemical that neutralizes the stomach acid. So it's coming out of that pyloric sphincter at pH 1 to 2 inches later, it's getting the sodium bicarbonate, which we saw in the last lecture is a buffer. It is an alkaline buffer by itself. It has an alkaline pH, and then it's buffering the stomach acid. Out of this 20 feet of small intestine, the duodenum is only the first 10 inches, and by the time it's done with the 10 inches, it's already slightly alkaline. So this is really, really important. In the first 10 inches of the small intestine, that chyme, and we still call this chyme, by the way, in the small intestine, goes from pH 1 to 2 to pH almost 8. The chyme is going to stimulate the small intestines to release cholecystokinin. We're going to see how that works here. Well, this is what it does. It's going to stimulate the pancreas to release the majority of digestive enzymes. It's going to cause the gallbladder to contract and squeeze out bile and bile salts. We'll talk about bile more later. What you want to write here next to this slide, and just make sure you have this down, the enzymes of the intestinal glands in the small intestine look very similar to the enzymes of the pancreas. They're close, they're not identical, and the pancreas is doing this much enzymes, and the intestinal glands are doing this much enzymes. You got to write next to the slide that the pancreas is the primary source of digestive enzymes, by far and away. Slide 23 is from your textbook, and it's showing you this connection between the secretin and the release of bicarbonate to neutralize the stomach acid within 10 inches. Okay, remember that the pancreas is a mixed gland. The vast majority of it is exocrine. How do I know it's exocrine? Because these digestive enzymes go into the pancreatic duct to the lumen of the duodenum. And the lumen is the body's exterior. And exocrine glands, like sweat glands, secrete into ducts to the body's exterior. So the vast majority of the pancreas is exocrine. The pancreatic islets are the parts that are endocrine. So here, when we're talking about the digestive system, we are talking about the exocrine function of the pancreas. With these digestive enzymes and the watery fluid rich with sodium bicarbonate. Now, if you look at these enzymes, and it says they're highlighted, primary source of exocrine digestive enzymes, it can digest all 4 biologically important macromolecules. Carbohydrates is the first diamond. I put lipids as the second diamond. Proteins are the third diamond. And even nuclease to break down nucleic acids. Again, you don't need to digest nucleic acids, but you will, because it's going to be there. If you're eating cells, they're going to be there. Your body says, why not? Now, if you look at the next slide, slide 25, it looks to be very similar. It's not. So on slide 25, write slightly different for the pancreas and much less. Intestinal glands do not secrete nearly the same volume of digestive enzymes as the pancreas. These are also slightly different in that when the amylase breaks down starch into disaccharides, the intestinal glands do produce the small amount of enzymes that break disaccharides into monosaccharides, sucrase, lactase, maltase. Some people, their intestinal glands can't make lactase, and some of them make it marginally, and then as the person gets older, they give up the ghost and stop making lactase. And what does that cause? Anybody?
Lactose intolerant?
That's it. That's it. Lactose intolerance. You can't break down lactose. If you can't break it down, your gut bacteria will break it down. And when your gut bacteria breaks it down, it causes gas at the best and other GI distress at worst. So it's easy to get around though. Okay, and then it has peptidases and then nucleases. But here's how it breaks down. Okay, this is the way you got to remember lecture exam 1 material on peptide bonds and proteins. Remember how amino acids are NCC, and the peptide bonds are between NCC links. I'm not going to draw all the covalent bonds. I'm only going to draw those peptide bonds. The amino side has two hydrogens, hydrogen, then the R group comes off the second or the middle carbon. This, by the way, is also the reason why proteins are buffers. You can always add in another hydrogen here, making it charged. You can take off a hydrogen here, making this side charged. And this is why proteins can either add in or take out hydrogen ions, why it's a good buffer. Anyway, so when you look at this slide on the bottom, you see that trypsin, let's see, I better do it this way. Trypsin is an enzyme that works on that end of this peptide. Pepsin is already broken down, a protein that's hundreds of amino acids long into, 5 to 10 amino acids. And then it becomes soluble. That's why the egg white is supposed to disappear. Well, trypsin is working on that peptide bond near the COOH end, the carboxyl end. Trypsin activates chymotrypsin and carboxypeptidase. And these other enzymes will break the other peptide bonds. But as it's doing it, trypsin is going from this end to this end, moving this direction, breaking this peptide bond and breaking this one. And eventually, you will have all individual linked amino acids. Okay. Slide 26 is showing you how this is happening. We have these enzymes coming from the pancreas, but you have to have the precursor to trypsin be activated by enterokinase. So trypsinogen, so even though all these precursors are coming from the pancreas, so I'm going to underline them. They're coming from the pancreas. This is coming from the intestinal glands. Okay. Okay, let's talk about absorption. That's the last content slide. All right, the reason why we're able to get the most absorption in the small intestine is because the pancreas secretions are coming in, and that's the primary source, is coming in at the very beginning of 20 feet of small intestine. Within 2 inches. Both the pancreas and the bile at the very beginning. And also, the pyloric sphincter is actually just squirting chyme out, and not in one big gush. It's going, and that's the controlled chyme release. So going into the stomach can be this huge, gluttonous, like big ass meal. You're just pouring it down. It can go into the stomach really big, but it comes out of the stomach really slowly. And if it comes out of the stomach slowly in controlled chyme release, you have more favorable surface area to volume ratios with the digestive enzymes. We learned about that in lab 3 as well. Okay. Then I'm going to expand on this after lecture exam 3 on Wednesday. So next Monday, I'll expand more on other things that is the reason why there's the most absorption in the small intestines. Okay? I think that's it. Let's go over the whiteboard here. Everybody got everything. Here is the left bank. Also, you don't have to worry about the cocospirometer mouthpiece because obviously you're not doing lab 14. This is the middle bank. But the words that were on the left bank, mechanical processing, secretion, digestion, absorption, that is all color-coded for what's on the whiteboard. And then that is continuous. I'm going to kind of slowly do this so you can see what is continuous on the right bank. And this is just showing you how the protein digestive enzymes interlock. Because trypsin is what activates these guys. It's what is responsible for activating chymotrypsin and carboxypeptidase. Okay, we're done a good 10 minutes early, which is what an introvert wants. By the way, if you were wondering what this is, that is the common medical chart abbreviation for alcohol, ethanol. Any questions? Since we're done early. Again, do not put things off to the end with online. You never know when there's going to be a glitch. And then that way too, you can let me know if there's a problem while I'm in my office and I can send out emails and find out what's going on and not wait till Sunday. Okay? No questions? Week 14 is done. Good luck on Wednesday.
Thank you.